My name is ADupert, and I will be going over the process of phagocytosis and inflammation. So we're going to dive into three key learning objectives. First, we're going to break down the cellular and molecular processes involved in phagocytosis and inflammation. Then we'll explore the specific molecular signals, like cytokines and opsocins, that guide these immune responses. Finally, we'll connect this to clinical conditions that arise when these processes malfunction. We're not just trying to understand what happens, but why it matters. So, innate immunity. The innate immune system acts as our immediate defense source. It's rapid and nonspecific, responding to a wide variety of pathogens without needing prior exposure. Key players include physical barriers such as the skin and mucous membranes, but also cellular elements like macrophages, neutrophils, and natural killer cells. The inflammatory process is one of its hallmark responses. Importantly, this system acts as a bridge to the adaptive immune response, particularly through antigen-presenting cells. So, phagocytosis. Phagocytosis is the process by which immune cells engulf and digest harmful microbes. Neutrophils are usually the first responders, followed by longer-lived macrophages and antigen-presenting dendritic cells. These cells act like surveillance and cleanup crews. They don't just destroy threats, they also gather intel. Dendritic cells, for example, will take microbial fragments to lymph nodes where they present these antigens to naive T cells, initiating adaptive immune memory. Phagocytes don't just wander blindly, they respond to specific chemical distress signals released by damaged tissues or invading microbes. These include interleukin 8 or IL-8 and complement protein CF5 or C5a, excuse me. Once they reach the area, the recognition step begins. Here, pattern recognition receptors like toll-like receptors or TLRs detect conserved microbial features such as lipopolysaccharides on bacteria. If the microbe has been marked for destruction by the immune system, it's often coated with opsocins or molecules that tag it for identification. Two key opsocins are IgG and C3b. IgG is an antibody, part of the adaptive immune response that specifically binds to antigens on the pathogen surface. This binding not only neutralizes the pathogen, but also flags it for phagocytosis. C3b, on the other hand, is a fragment of the complement protein C3 that covalently attaches to microbial surfaces. It acts like molecular glue that enhances phagocyte binding through complement receptors. This dual tagging system, antibodies from adaptive immunity and complement from innate, ensures efficient recognition and ingestion of pathogens. Think of IgG and C3b as high visibility jackets for invaders. They make the pathogens stand out, so the immune cells know exactly who to target. So once a pathogen has been recognized, the phagocyte extends pseudopods, or these arm-like projections that you see here, to surround and engulf it. These arms fuse to enclose the pathogen in a vesicle called a phagosome. This step, very important, as it isolates the pathogen within a membrane-bound compartment. The actin cytoskeleton plays a very important role in reshaping the membrane, ensuring a tight seal around the invader. It's a bit like a cellular Ziploc bag. It keeps the contents enclosed for processing. Step 3. The phagosome then undergoes fusion with the lysosome to form a phagolysosome, creating a highly hostile environment for the pathogen. Inside, digestive enzymes such as proteases and lipases work in tandem with reactive oxygen species, or ROS, as you can see up there, like superoxide and hydrogen peroxide, to destroy the microbe. This dual chemical and enzymatic assault ensures the efficient breakdown of pathogens. However, some microbes, like Mycobacterium tuberculosis, resist the process by inhibiting phagosome-lysosome fusion. After the pathogen is degraded, the immune system doesn't just dispose of the remnants, it uses them. Pieces of the pathogen or antigens are loaded onto major histocompatibility complex, or MHC, class 2 molecules, and displayed on the surface of the phagocyte. And you can see that with the illustration up there. This is especially important in dendritic cells and macrophages, which act as antigen-presenting cells, or APCs. These antigens serve as warning signs for the T cells, triggering adaptive immune responses that are more targeted and long-lasting. Now that we've explored how phagocytes recognize, engulf, and destroy harmful invaders through phagocytosis, it's important to understand how this process ties directly into the broader immune response, particularly inflammation. While phagocytosis is the more hands-on cleanup and defense mechanism, inflammation sets the stage for that response to happen in the first place. In fact, the very act of phagocytosis helps amplify and shape the inflammatory response. As phagocytes engage with pathogens, they release chemical messengers that signal other immune cells to join the fight. This creates the redness, warmth, swelling, and sometimes pain we associate with inflammation. So, in a sense, phagocytosis doesn't just respond to inflammation, it helps drive it. So now we're going to look into how inflammation unfolds and why it plays such an important role in both launching and resolving the body's immune response. Okay. Inflammation. It is a defensive response to infection or tissue injury. Inflammation begins with vascular changes. So the blood vessels dilate, or vasodilation, increasing blood flow, causing the redness and warmth associated with inflammation. Simultaneously, the permeability of blood vessels increases, allowing plasma proteins and leukocytes to leave the bloodstream and into the tissues. This leads to edema, which you've probably heard before, or fluid accumulation, and this contributes to the swelling and discomfort that we know. These changes are driven by mediators like histamine, prostaglandins, and nitric oxide. Cytokines and chemokines released at the site of infection act like homing signals, drawing neutrophils and monocytes into the tissue. These undergo diapedesis, or the squeezing through gaps in the endothelial lining of the blood vessels, like you see there. Once inside the tissue, they follow chemotactic gradients to reach the source of infection. It's a highly coordinated migration, and any disruption can impair immune response. Neutrophils often arrive first, delivering the quick punch, while monocytes follow to clean up the repair. Resolution phase. The resolution phase is actively regulated by anti-inflammatory signals such as IL-10 and transforming growth factor beta, or TGF-beta. Macrophages begin clearing apoptotic cells. I've always heard my high school science teacher always said apoptosis, but apoptotic cells and debris. Macrophages begin clearing apoptotic cells and debris. How do you say it? A process known as phagocytosis. If these processes fail, unresolved inflammation may transition into chronic inflammation, which can drive disease processes, including autoimmunity and fibrosis. Resolution is not passive, it's an intentional shift towards healing and restoring tissue homeostasis. And here are some examples of the normal tissue and the fibrotic tissue. And these persistent inflammation that we talked about resulting in the fibrosis and granuloma formation, we're going to talk about in just a second with some clinical cases. And some conditions that could arise from this is the rheumatoid arthritis and Crohn's disease. So let's get into one of these examples of the granuloma formation. So chronic granulomatous disease, or CGD, we're going to stick to that, is an inherited disorder where phagocytes lack the NADPH oxidase complex required to generate the ROS that we talked about earlier. As a result, they can engulf pathogens, but they can't destroy them. This leads to persistent infections and granuloma formation, where immune cells surround the microbe but can't eliminate it. Patients suffer from recurrent infections, and granulomas may cause obstruction in organs like the GI tract. This case underscores how essential the ROS are to proper phagocyte function. Autoimmune diseases like rheumatoid arthritis or lupus involve the immune system attacking self-tissues. Here, chronic inflammation is driven by cytokines like TNF-alpha, IL-6, and IL-1 beta. These mediators promote tissue destruction, particularly in joints or connective tissue. Biologic therapies, such as monoclonal antibodies against TNF-alpha, aim to suppress this inflammation and preserve tissue function. These therapies highlight the precision possible when we understand the immune signaling. And these immune checkpoints, I found this cute little cartoon. It's adorable. So, to wrap up, phagocytosis and inflammation are indispensable for host defense, but their balance is very important. Too little leads to infection. Too much causes tissue damage. So some things to think about as you walk away from this. Let's discussion questions. Why are abscessins like IgG and C3b so essential to effective immune clearance? How might therapies that enhance phagocytosis benefit chronic inflammation conditions? Could better understanding of phagosome maturation offer strategies for antimicrobial treatment? Just some things to consider and discuss in our discussion questions as I have to post this. Thank you for your attention, and let's discuss. And I have my references here. Thank you so much for listening. I wish you the best discussion.